Hello everyone, welcome back to the class. In our previous class we had discussed about anaphase stage of mitosis cell division, which is the shortest phase of mitosis. Each sister chromatids of the metaphase chromosome is attached to the kinetochore microtubule via its kinetochore, and the opposite ends of these kinetochore microtubules associate with one of the spindle poles. At this stage, that is anaphase stage, the kinetochore of microtubule pull the two kinetochore towards the opposite poles. Sister chromatids do not separate because they are held together at their centromeres by multiprotein complex called cohesin. This cohesin degrades, and before that degradation, cohesin will be associated with the chromatin at S phase itself. During this phase, the separase degrades the cohesin. Before anaphase, a securin protein keeps separase in an inactive state. This is anaphase at the beginning stage and the ending stage. So sister chromatids do not separate because they are held together at their centromere by multiprotein complex called cohesin, right? How is this cohesin degraded? It is because of APC bar C. MCDK activates APC bar C. APC bar C functions in coordination with two homologous mitotic coactivators CDC20 and CDH1. APC bar C activated one initiates the ubiquitination and degradation of securin and releases the separase. Free separase cleaves cohesin complex. After separation, each chromatid, which is now called as a chromosome, is pulled slowly towards the pole it faces. Newly separated chromosomes move at same speed. Chromosomes move by two independent and overlapping processes. Anaphase is divided into two phases, that is anaphase 1 and anaphase A and anaphase B. Anaphase A is the initial poleward movement of the chromosomes, which is accompanied by shortening of the kinetochore microtubule. The force generated by microtubule is by depolymerization at the kinetochore, which there is a loss of tubulin subunits at the plus end as kinetochore move towards the pole. Movement towards the spindle poles are facilitated by kinesin 7 and kinesin 13 motors that are localized at the kinetochore. Kinesin 13 proteins, that is which do not have the motor activity, while in anaphase B, which depends on motor proteins at the poles and that pull the poles apart, as well as motor proteins at the central spindle that pushes the poles apart. The two poles move farther apart. It happens due to the sliding of polar microtubules past one another. Plus and directed kinesin 5 motor proteins cross-link adjacent overlapping antiparallel polar microtubules and slide the microtubules past each other, thereby pushing the spindle poles apart. There is a dynein motor which anchors astral microtubule plus ends to the cell cortex, pull the poles apart. So this was about anaphase. So now in today's class we will learn about the last stage of mitosis cell division, followed by another process which is very much important for the division of the cytoplasm, that is cytokinesis. Telophase stage and cytokinesis of mitosis cell division. So the question arises, what happens in telophase? So in telophase, telos means end. The word telo or telos means end. The separated daughter chromosome which arrives at the pole and the kinetochore microtubules disappear. This kinetochore microtubule disappears. The polar microtubule elongates still more and a new nuclear envelope will reform around each group of the daughter chromosome. The condensed chromatin expands once more. The nuclei which had disappeared at prophase will start to reappear, that is nucleoli, and the mitosis is at an end. So there is nothing much important in telophase. It's just a separation and it's just a reformation of nuclear envelope. See here, telophase, which is the separated daughter chromosomes, arrives at the poles and the kinetochore microtubule disappear. The polar microtubules elongate still more and a new nuclear envelope reforms around each group of daughter chromosomes. The condensed chromatin expands once more. The nucleoli, which had disappeared at prophase, begins to reappear. Followed by this telophase, there is another important phase or a process, that is cytokinesis. Cytokinesis. This cytokinesis is nothing but the division of the cytoplasm. Division of cytoplasm is known as cytokinesis. In a typical cell, the cytokinesis is associated after, or it is accompanied, it accompanies every karyokinesis. So first the nucleus will divide and then cytokinesis will happen. First the nucleus divides and at last cytoplasm divides to form two daughter cells. But in certain organisms, if cytokinesis accompanies with karyokinesis, it will form two daughter cells or equal daughter cells, we may say, and uninucleate. Majority of them will form uninucleate, means in a single cell, single nucleus, single cell, single nucleus. But in some certain organisms, this cytokinesis, after cytokinesis, it does not follow karyokinesis, which ends up leading to multinucleate cells. Here in this, the cytokinesis does not follow karyokinesis and there is a repeated nuclear division which lead to multinucleate cells. The cytokinesis begins in anaphase. So it begins in anaphase and ends in telophase. The mechanism of the cytokinesis in animal cells and plant cells, they vary. So we can differentiate like this: in animal cell and in plant cell. In animal cell, the cytokinesis mechanism is achieved by cleavage, while in plant cell the cytokinesis takes place by the formation of cell plate. So in animal cells it is by cleavage and in plant cells it is by cell plate formation. Let us further know about these different types of cytokinesis. First, let us see the cytokinesis in animal cell. In animal cell, what happens here? We mentioned that in animal cell the cytokinesis is achieved by cleavage. Cytokinesis appears to be guided by organized bundles of actin filaments. So this actin filaments plays an important role in cytokinesis, which is guided by organized bundles. Cytokinesis is guided by organized bundles of actin filament. So during cytokinesis, the cytoplasm divides by a process of cleavage. So this cleavage is accomplished by contraction of circumferentially oriented contractile ring. So remember this contractile ring. So this contractile ring is made up of actin filament as well as myosin 2. Where did we started from? So cytokinesis in animal cells is appear to be guided by organized bundle of actin filaments. So during cytokinesis, the cleavage is accomplished. This cleavage is done is accompanied by contraction of circumferentially means surrounding the outer region. So circumferentially oriented contractile ring. So contractile ring is made up of actin filament and myosin 2. Where are these bound to? So these are bound to cytoplasmic phase of plasma membrane. So this contractile ring defines the cleavage furrow. It defines the cleavage furrow. It acts very much in this cleavage process. So the muscle, like the sliding of actin and myosin 2 filaments in the contractile ring, generates a force. So contractile ring will generate a force required for cleavage. And another important factor is that RHOA. So RHOA or Rho A is a small GT pace of RAS superfamily. This controls assembly and functioning of contractile ring at the site of the cleavage. So Rho A is activated at the cell cortex at the future division site, where it promotes the actin filament. So after activation, it promotes actin filament by activating formin. To write like this, first Rho A will be activated at the cell cortex. Activated Rho A activates formin. What happens if formin is activated? It will lead to assembly of actin filament. I'll write it as actin assembly. What does the activated formin does? It promotes actin filament assembly. The myosin 2 assembly and contraction require other protein kinase. So this Rho is to activate formin followed by actin assembly. So what about myosin? So it is activated by ROCK, that is Rho activated kinase. Okay, this kinase, ROCK kinase, is nothing but it phosphorylates. So it phosphorylates the regulatory myosin side chain or light chain, it phosphorylates the myosin light chain. What happens if there is phosphorylation of myosin light chain? It promotes assembly and contraction. Assembly and contraction of actin myosin ring. Okay, so this is cytokinesis in animal cell. Let us know further about this animal cell. So in anaphase stage, so there is a pole, there are some tubules, microtubules, and we have separated chromosomes now. So as I said, so there is a contractile ring which is circumferentially oriented. What do I mean? It's like this. So it is circumferentially, it is outer. Okay, it is bound to the cytoplasmic phase. So this contractile ring is made up of actin filament plus myosin 2. So this picture is of anaphase stage. Now what happens in late telophase is that the contraction begins. The contraction of the contractile ring begin with the help of rho A and rock. So this contraction begins. Now the cell will contract at the middle and the nucleus forms. The nucleolus which was disappeared will reappear, and there is pole also. So this picture is in late telophase. Okay, this contractile ring appears to be condensed or contracted. It is due to activation of rho A and activation of rock, which in turn activates a series of reaction. So this was about animal cells cytokinesis. Now let us just recall what did we learn now. Cytokinesis is a division of cytoplasm. Cytokinesis accompanies every karyokinesis. Cytokinesis begins in anaphase and ends in telophase. The mechanism of cytokinesis in animal cells and plant cells vary. In animal cell, the cytokinesis is achieved by cleavage, and in plant cell it takes place by cell plate formation. Cytokinesis in animal cell. So cytokinesis is guided by which bundle? Bundles of actin filament. Cleavage is accomplished by contraction of circumferentially oriented contractile ring, which is made up of actin filament and myosin 2 bound to the cytoplasmic phase of the plasma membrane. So it defines the cleavage furrow. Okay. Muscle, like the sliding of actin and myosin 2 filaments in the contractile ring, generates the force required for cleavage. So here you can see actin filament, bipolar myosin filament, which is contributing to contraction of contractile ring. See here, rho A. How does this help in assembly? Rho A is what is in GDP stage, that is GDP state, which is inactive. So with the help of rho Jeff, it is activated, it becomes rho A GTP. When GTP comes and binds, it becomes activated. It activates formin, which initiates actin filament formation. It also activates rho activated kinase, that is ROCK. It phosphorylates the regulatory myosin light chain, where it leads to myosin 2 activation. Both this activated forms the assembly and contraction, responsible for assembly and contraction of actin myosin ring. Now let us know about cytokinesis in plant cell. As we know, cytokinesis in plant cell is accomplished by cytokinesis in plant cell is accomplished by cell plate formation. Cell plate formation. As there is a rigid cell wall, in plant cell, it is difficult for the cytoplasm to divide. Hence, instead of forming a furrow, the cytoplasm is partitioned by construction of cell plate inside the cell. The cytoskeleton framework, here comes the importance of cytoskeleton. So this framework determines the plane at which the plant cell to be divided, whether here or here. So this the plane of plant cell division is determined by cytoskeleton framework. We knew that plant cell is having a rigid cell wall. So now array of microtubules, so it all starts from G2 phase. So an array of microtubules appear in G2 phase, and this microtubules form ring around the entire cell just beneath the plasma membrane. Okay, or I may write like this itself. Let me change the shape. If this is a plant cell and it is having a plasma membrane inside, microtubules form a ring around the entire cell just beneath the plasma membrane. So it is called a pre-prophase band. So array of microtubules appears in the G2 phase, and it forms a ring around the entire cell. That ring is known as pre-prophase band. So this also contains actin filament in addition to microtubule. It also contains what? Actin filament. So the microtubule disappears in the late anaphase stage. Okay, in the anaphase stage, the microtubule disappears before metaphase stage, but the actin filaments remain and provide a memory of the predetermined division plane. So to represent it clearly, I'll write a plant cell here which is having a plasma membrane, and there is already separation completed. Now it's time for cytokinesis. So these are polar microtubules. So this is in anaphase stage. So in late telophase stage, what happens is, so again this is a single plant cell having a plasma membrane. It forms a pre-prophase ring around the entire cell just beneath the plasma membrane. So we know now that the microtubule disappears, but there are actin filaments which remain and provide the predetermined division plane. Okay, in the late anaphase stage, an array of microtubules together with actin filaments. So now actin filaments and microtubules come together, they assemble in this plane and forms a cylinder-like structure around the cell. That cylinder-like structure is known as phragmoplast. What is phragmoplast? It is the array of microtubule together with actin filament. Microtubule plus actin filament forms a cylinder-like structure which is known as phragmoplast. Along with this, there is another one key player, that is small membrane bound vesicles. So these small membrane bound vesicles is derived from Golgi apparatus, and they are filled with, this is a very important thing, they are filled with cell wall precursor, which fuse with the phragmoplast. So this fuses with the phragmoplast and forms the early cell plate. So the polysaccharide molecule derived from these vesicles forms the matrix polysaccharide. Sometimes later cellulose microfibrils are also laid down within the cell. So this forms a cell and this cleaves the cell. This becomes cell 1 and this becomes cell 2. Now let us see the image. Cytokinesis in plant cell, it differs from animal cell because the presence of rigid cell wall. Instead of forming cleavage furrow, cytoplasm is partitioned by the construction of what? Cell plate. So this must be cell plate. Mistakenly it has become late. Sorry for the mistake. Cell plate inside the cell. Cytoskeleton framework determines the plane of plate division. Array of microtubule appears in the G2 phase itself. It forms a ring. That ring is known as pre-prophase band. It also contains actin filament. Microtubule plus actin filament forms pre-prophase band. Microtubules disappear before metaphase. This and actin remains and provide the memory for predetermined plane. In late anaphase stage, an array of microtubule together with actin filaments assemble in this plane and forms a cylinder-like structure called phragmoplast. Along with that, small membrane-bound vesicles derived from Golgi apparatus are filled with cell wall precursor, fuse with the phragmoplast and form the early cell plate. Becomes the polysaccharide molecules delivered by these vesicles form the matrix polysaccharide. Later, cellulose microfibrils are laid down within the cell plate to complete the new cell wall. So this is another image showing the new cell wall formation. See here, new cell wall formation by the cell wall precursor, which is released by membrane bound vesicles derived from the Golgi apparatus once it fuses with the phragmoplast. So this is the formation of cell plate between two daughter plant nuclei during cytokinesis. So there is a small topic after this, that is syncytium and cenocyte. You can make out the difference in this image. So syncytium. So in some organism, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises. So here nuclear division is completely done, but cytokinesis is missing. So therefore it results in multinucleate condition or multinucleate cell, where in a single cell many nuclei are present. So this is known as cenocyte. Example for this is the blastoderm of the Drosophila. Syncytium is dead opposite for this cenocyte. So it is a multinucleate cell which forms as it is a multinucleate cell. How it is formed? It is formed as a result of fusion of multiple uninucleate. When they fuse with each other, they form a multinucleate cell. Example is formation of skeletal muscle. So this is the major difference between these two. So that's all for today. In our next class, we'll start the second type of cell division, that is meiosis. Thank you.